So I just want to start the question for the panelists, kind of reflect the year just passed, 2018, and how does each panelist feel about the year 2018, how industry collectively advanced, and how do you feel about the 59 new drug approval? Do you proud of it, or you still think with the industry collectively we have not done enough? Mike, start from you.
Yeah, I guess I would say I'm proud of it. Both from a company perspective in terms of Roche and Genentech, but also from an industry perspective. I think that, to me, a little bit of what you are seeing is kind of the reality of the FDA changing their persona. I mean, they have changed the way that they interface or interact with sponsor companies, and have changed themselves from people who throw hurdles in front of companies to people who facilitate moving past those hurdles. And so for me, I think it's very positive. And I hope that some of those behaviors begin to spread into other therapeutic areas outside of, say, oncology and HIV. And so, yeah, I'm quite encouraged. Good number. And, let's keep pushing the number and keep pushing the FDA to be a facilitator in this business.
Thank you. Dr. Mammen?
Yeah, it's hard to say anything, but we're highly pleased with the performance of the entire industry. But I think it's important just to hold that in check. We should measure ourselves not by the number of drugs, but by the totality of the medical impact that we're able to have. And then you showed a slide at around the number of trials that it's taking in order to get that level of approval, the amount of investment that's happening to get those number of approvals or medical impact. And I think that we have a long ways to go, and we can talk about that a little bit further in the conversation.
So, Laurie?
So I think 2018 was an excellent year. Out of the 59 drugs, 17 were cancer-related. I also think that I'm encouraged by Scott Gottlieb's recent announcement, I think today, that he's going to reorganize the FDA and make a special subgroup of individuals who will be there to review new drugs and do some of the precision medicine testing and patient response and get that data and analyze that data. So I think that Scott Gottlieb is doing a really good job in moving us to the next stage. Now, you asked how many drugs are going to be expected for 2019, and I would say that since the government is shut down, including the FDA, with no obvious hope of opening up anytime soon, that we may have a few fewer drug approvals. I hope not.
Oh, God.
But as you said, I think it was you, Michael, who said it's the quality of the drugs that were approved in 2018 that was particularly remarkable. Very high quality, not just quantity, but a lot of drugs in new classes, occupying new chemical space.
Thank you.
Yeah, so I'd like to, I find that very exciting, and as a former head of R&D of a large pharmaceutical company, I'm acutely aware of the fact that the medicines approved in 2018 are the fruit of probably the past decade of progress and work in science, and that's a very good sign for the future. But it's also a signal to those of us who are pessimistic vis-a-vis the industry, as many were and are, but particularly were, let's say, in the early 2000s, that this industry has a very long turnaround time between an idea or a technology and the fruits of that as an approved medicine. So what we're seeing now is probably the beginning of a new wave, and clearly the progress and the changes in the FDA are enabling it. But it also, I think, comes with a challenge, which is an incredible amount of redundancy into the system that I'll be commenting on further, because these are the approved medicines. It will be very interesting to ask the question, how many other drugs against the same targets are among the 250,000 clinical trials being tested?
Okay.
and what will happen with them.
But notably, in the 2018 new drug class, a lot of those are for very specific biomarker-driven or rare diseases or orphan drug diseases. So they are targeted to small patient populations. So it's somehow the larger indications like cardiovascular, diabetes, those kind of is not all exist. Don't you think the industry right now is kind of pursue more, I would say, smaller size clinical trial, relatively certain because the biomarker-driven or certain genetic mutation-driven tumor or other diseases. So the success rate is actually higher than pursue chronic like Alzheimer's disease, Parkinson's, or the major disease out there today has no treatment. Anyone want to comment on that?
Well, I guess what you're seeing is the incentives in the system, right? There are incentives that have been put into the system for rare diseases and orphan diseases and those types of things, and then in areas like oncology. So in some respects, it isn't a big surprise that you see the bulk of the work in industry going on in those spaces where they are incentivized to work in them. I'm not entirely sure that I know of any incentives for people to work in metabolism, from a government or a regulatory perspective, right? And then, of course, there's all the risks that you stack on top in those disease areas, particularly from a safety perspective.
Cancer, of course, is one of the areas that's being focused on. In particular, not just cancer genomics and not just CAR T cells, but also cancer immunotherapy as a whole. And we feel like we've done such a great job, right? And there's no question that a decade and a half ago, who would have predicted that we would actually be able to treat people successfully with immunotherapy? And yet, it's really important to remember that we're only treating 20% of patients with immunotherapy. And there are and only 8 or 9 tumors are sensitive to immunotherapy. So we have a very long ways to go, both in cancer genomics and in cancer immunotherapy. And what I find a little bit disturbing is, from what I know, the major pharma companies are focusing on the same thing, okay? So we want more checkpoint blockers. We've got to get more checkpoint blockers. We only have 2, right? We may have 31 molecules against PD-1, which I think is an enormous waste of time, as you pointed out, Ed. But we gotta be exploring other spaces. And I think the best way to do that, it's not gonna happen in big pharma. It's gonna happen in academia and in small biotech. And I'll say more later about what I think the interaction between those entities should be.
So I'm gonna follow up with you. Maybe this is a good time, Laurie, I follow up a question for you. Is recently Dana-Farber and Deerfield actually created a protein degradation institute. The funding coming from Deerfield is a Wall Street investment firm. It's very unusual. And so can you share your thoughts why you try this kind of arrangement or collaboration in search for breakthrough therapies in the field of protein degradation?
So, a number of reasons. Deerfield has got a very unique approach to funding science at academic institutions. And that is, they're generous about the potential outcome and who gets compensated for what. So, I believe most of the key discovery of new targets is going to take place in academia, but we can only get it to a certain point. And we get it to, at Dana-Farber, we can de-risk it quite intensively because we have so many pre-genetic clinical models. We have ex vivo organoid all-in-human systems. We got a lot of new technology that will allow us to predict who's going to respond to what in terms of how many clinical trials you have to do. So we can increase the probability of success at Dana-Farber, probably from 1 out of 10 in the industry in terms of filing INDs to about 1 out of 4. But what do we get for it? Academic medical centers are under huge threat, right? We have declining healthcare reimbursements. We have inadequate funding from the government. And yet we have brilliant researchers with many ideas who need support. And what Deerfield has recognized is that they will put the support in the basic and translational science. So the agreement with Deerfield will provide up to $80 million to fund a multiple series of projects in the protein degradation field. And just to say a word about protein degradation, you know, there are many targets that have been considered to be undruggable, whether they're transcription factors or they just don't have any nice clefts in which you can fit a small molecule. But you can develop protein degradation technology, which has been developed by Dana-Farber and by Yale. Those are the two major academic institutions that have been leaders in the field. And that allows the universe of targets to expand exponentially. But Deerfield recognizes that they have to share the benefits with academia. So we have 50% equity. Now, the usual equity that we get from working with VC firms or pharma, maybe 2% royalties because we partner at a fairly early stage. If we want to really exponentially increase the number of new drugs, which means new targets, then pharma's got to work with academic institutions. And it has to reward them for the brilliant discovery work they do at whatever stage they decide to partner.
Okay.
Because to me, a marriage between academic institutions and the private sector is a marriage made in heaven for our patients.
Thank you very much. Very good point. So I'm actually turning to Dr. Mammen and also Mike. Given what Laurie just said about academic and investment collaboration, how in your large big pharma R&D organization pursue in terms of enhanced R&D productivity, generating more new target drugs? Do you have any specific thoughts on it?
I can start. You know, I think of this as a lot less clear-cut than I think Laurie might imply. I think if you have very motivated people with great, clear ideas, clear thinking, and a willingness to go out on a limb a little bit, you can actually discover in a lot of different places. And I applaud anyone that tries, because this is ridiculously hard, what we do. And I think that for a company the scale of some of the companies that are on this panel and in our industry, you can't be too picky about one model. So for me, it's beyond even the redundancy within oncology, Ed, that you referred to. We have to be inventive for the full spectrum of medicine, right? Full spectrum of medicine, and we, you know, at least at my company, we're agnostic as to the source of the idea. We'll work with, if it happens to come from an internal lab, great. If it happens to come from a postdoc in an academic setting, fantastic. If it happens to come from a biotech company, amazing, it's excellent. And if we can add value to the progression to see that drug through to patients, great. And so this is a really an agnostic approach, and I think we do our best to try to plant seeds. We try to do our best to plant seeds in as many places as possible, and work to make ourselves as muscular as possible in the areas I think that we are definitely insightful and good. And together, I think we'll get some good medicines made.
Yeah, I guess what I was going to say, just to add that certainly in our perspective, the academic community is absolutely inextricably essential to the healthcare system, full stop, right? And that's how we think about it. So we have a very active partnering program. We have enormous numbers of academic collaborations in our organization. We have a postdoc program that has 120 postdocs in it. And that is done for a whole host of reasons, but one of them is that it keeps us connected. These postdocs do basic research. They do not work on project teams. They do basic biological, mostly research, and that keeps us very plugged into the academic community in terms of what is going on scientifically, what are the opportunities, what are new targets, and those types of things. I guess I would add, new targets, you know, we are totally indifferent to where new targets come from or where molecules come from. What we want is what is the best for patients. And new targets come from many places, academics being one of them.
So, Moncef, you previously you ran a very large, also a big pharma R&D organization. Now you are investing into the emerging biotech company, technology companies. In your view, certainly when you invest in small company, you hope they're more agile and then more creative and more driven. In terms of go after number of things, in your mind, what are the things you feel most important for venture startup companies being successful?
Yeah, so I mean, let me first say that I see what we do as an ecosystem with many different players, and it's a story of and, not of or. However, there is definitely a need to rebalance the power and the distribution of reward within the players of that ecosystem. A piece of it, the payer piece, has substantially changed. The discovery piece, the upfront piece of it, maybe in that process, and in that area, the academic community, of course, and also the biotech community, which very often stems from the academic community, are playing a bigger and bigger role. And it's like very important to stress that 23 new medicines approved in 2018 were approved, discovered and developed and delivered from within smaller companies. I think for me, over the last 18 months, moving from a large R&D role where you are an investor, effectively, and an allocator of capital to a venture capital allocator, one of the biggest differences is actually attention to the cost of failure. It's really interesting that the cost of success is, when you look back, is not what really makes a difference. What strikes me as I get into the guts of the venture capital world is in particular, the one I joined, but I think in general, is the attention to the cost of failure. So in my previous life, on January 1st of every year, anywhere between 60 and 65% of my yearly budget, which was multi-billions of dollars, was spent before you wake up in the morning, because there is an incredible fixed cost and infrastructure that's there, whether you discover the medicine or not. And next to it is also a number of ongoing clinical trials that take another 10 or 15% of that budget. At the end of the day, you really have 10% of that budget or something like that, a very small minority that you can be flexible with. In the world of venture capital, if appropriately managed, I would say two things. First, an overwhelming majority of the investment, the capital allocated, is allocated to the project versus to an infrastructure or to an organization, which immediately allows for a very high flexibility in terms of decision making and choices. And secondly, very importantly, because you don't have that fixed assets that are there, you don't have all these sophisticated machineries and capacity to sequence everything that moves and all, etc., you are less prone to ask more questions than you should, or at least ask questions that are not on the critical path to lead you to decide, does this project goes or doesn't go. And I think for those two reasons, my experience now is that the cost of failure is very substantially lower than it is in a very large organization. It's something to reflect on. The second thing I think is the level of redundancy. The level of redundancy in the pipelines, and I'd like to maybe later on discuss it in the pipelines of large companies, but also in the drug pipeline as a whole in the ecosystem.
Okay, overall, I think the industry's success rates are still not improved dramatically. So every drug entering into phase 1 trial probably only have 10% chance get a final approval, right? So that success rate has not really been improved. But if you look at individual therapies, particularly like a gene therapy, monogenic gene therapy, as long as it's safe, you pretty much have very high probability actually the drug will be successful. So when we are working on a new target, new research, I would ask actually Dr. Mammen and Mike comment on how your organization actually will go about discover new drug, whether you go by disease pathway or you're really looking for mechanism-based target validation. So in the end of the day, the disease and the target you're working on has higher correlation. So you have a high chance of success, regardless of what kind of modality you're using, whether small molecule, biologics, or cell gene therapy.
I guess so. I was going to say that biological insights is a big driver. I mean, and so the more insight you have, the better targets you're going to pick. I always say, okay, in one company that doesn't do good biology, they got 3 targets and they staff them all. You know, and a company that does good biology has 1 target and they staff that with 3 times as many people, right? And so biological insights are key to picking the right target in the end and understanding the relationship between that target and disease, and then understanding the relationship between the modulation of that target and the modulation of disease. And so certainly doing hardcore biology and dedicating the time is incredibly important. What I would say is that the approach that we take is generally a pathway approach. Right, we try to be agnostic about molecule type. So we take a pathway approach, we work pathways, we relate those pathways to disease, and along the way, the targets ultimately, kind of come along with that approach, but then you have to sort the targets. Sometimes you do that with molecules, sometimes you do that with biological tools, both of them are complementary. But yeah, we generally take a pathway approach and a mechanistic or a drug platform agnostic approach.
I think that that's incredibly important, right? And that's why the academic medical centers are so important, at least the good ones. Because, 80% of the data that's published is not reproducible when it's turned over to biotech or pharma. So you gotta align yourself with a place that can do extensive validation in the mechanism. The entire mechanism needs to be known, all the signaling pathways needs to be tested not only in vitro systems, it needs to be tested in a whole range of genetic models, whether they're PDX models or GEM models or whatever. And then at Dana-Farber we have a first-in-class, best chemical biology program, which is why Deerfield wanted to partner with us, partner with our chemists. So we're able to make tool compounds against those targets and test them in preclinical models. And that's why I say that the probability of success from a place like Dana-Farber, that has all of those components, is going to be higher. Because we have to be able to predict in the future, and I'll say more about this in a minute, we've got to be able to predict in the future what drugs to put a patient on and what clinical trials to do, because we cannot do thousands and thousands and thousands of clinical trials. At Dana-Farber we do over 800 a year, but we focus on early phase trials.
I'd like to pick up on that, because I think picking up on that and answering your question, one of the opportunities we have as an industry is to decide what denotes a disease. Right, oncology, I feel is like leading the way in some ways on redefinition, reclassification of disease. And as Mike said, if going one step past having a pathway mindset is understanding how that pathway, might influence a set of undesirable human phenotypes that may or may not have a known disease name today. Only this industry can actually push in that direction. It's not going to be the practicing physicians, it's not going to be the regulators on their own, but only the industry with academic medicine can push in this direction. So, the aspiration, as I said before, we shouldn't measure ourselves by the number of approved drugs. We shouldn't do that. We should look at the totality of the medical impact. So, to have an approved drug with a 30% response rate is okay, but that means there's a significant amount of opportunity there. Like, why not aspire, why not dream of a drug having 100% deep impact? And it really is around connecting that cloud of mechanism and being able to connect it to the cloud of things that we call diseases, but pick out the new boundary that is the signature, the measurement, the biomarker for full deep response to a particular molecule.
Can I just comment on that? I mean, there's redundancies in the system that makes that very difficult, what you just described. In some instances, there's a single driver. In most instances, it's way more complex than that.
You know, what turned HIV/AIDS into, from a lethal disease into a manageable disease, so that now a 20-year-old man who gets infected with the virus has a normal lifespan, is the idea of combination therapy. And certainly that's the answer for cancer. May not be the answer for everything, but to increase 20% to 30% to 40%, you got to figure out what other angles, what other mechanistic pathways are involved that lead to the lack of response. And that takes an enormous amount of data analysis. So, you know, one of the things that we do at Dana-Farber is patient comes in, take a biopsy. Biopsy has tumor cells in it, but it's also surrounded by a lot of innate and adaptive immune cells that are highly immunosuppressive. So we do whole exome sequencing, not only of the tumor, but of the tumor microenvironment. You take those huge buckets of data, you do that on enough patients, you add in machine learning for pathology, machine learning for radiology and biomarkers. You connect that to the medical chart, which you have now structured so that you can develop algorithms. You take all that 5 massive buckets of data, and I think that 5 years from now, we're going to be able to predict when a patient first comes in what combination of drugs to use on that patient based on those massive buckets of data. We have the technology now. AI is there. It should be possible to do this because we can't waste the precious time of a patient who comes in with cancer putting him or her on a drug that A, might not be efficacious, and B, might create serious toxicity, like some of the immunotherapeutics.
Well, I was just going to agree with you 100% and basically just comment that your description was the reason why we bought FMI in Flatiron.
Great. So that's another question from the audience, from pre-submitted questions. Is really asking for how do the advanced technologies, particularly AI, machine learning, will be factored into your potential success and also enhance productivity and predictivity for new drug discovery. Comment?
Yeah, I can start, but I think we're all thinking about this pretty hard right now. It means different things, different parts of the process of going from idea all the way to providing access to the right patients in the marketplace. Data analytics. As an example, you know, the multiple groups are right now thinking about how to use all the medicine that's practiced every single day and recorded in electronic health records to pull out from that patients that you would have put in a trial but you did not, the so-called concurrent synthetic control. But this is not necessarily like falling off a log. There are significant factors to kind of account for within there. If you're dealing with a medicine where it's used in a hospital and it's very likely that there are no skip doses, that's one thing. If you're dealing with a medicine that's in the real world where people might forget or for a number of reasons why adherence may be low, that's an entirely different thing. And so pulling out, for example, a synthetic control arm from the latter kind of situation is not quite right, because it's comparing apples to oranges. Or the nature of patients that are in a clinical trial sometimes are just a little bit different. Sometimes there are systematic biases from where you're drawing your data. So I'm optimistic, though. I think that this kind of thing can absolutely be done using real-world evidence to make trials better. But I think we just need to be cognizant of the particular challenges that might be present.
Yeah, I just wanted to comment on this a little bit and maybe focus my attention more on the drug discovery in the early part of the game. And I think that AI, to me, in drug discovery, feels a little bit like it did back in the late 1980s with structure-based drug design, in the sense that this was a technology that was unproven but felt like a step function if you could do it. And so I would put AI into that category. And so now structure-based drug design is done by everybody, and it's a standard tool that you use when you have the right protein in the right situation, right? Now, of course, what that requires, though, is to do that is you need structured data sets. Now, most discovery organizations in pharma have structured data sets, particularly in the small molecule arena. So this is where you will see this begin, and it has already begun in there. Now, I did kind of feel compelled to talk about some of the concerns, right? And the biggest one to me is that AI tools have inherent biases that are built in them because they only know the data that you showed them. And so if you say, I want to make a molecule that's a good molecule that looks like my old molecules, then AI is a great tool because you will have trained them on your old molecules, and then that will be the thing that the tool looks for, right? All right, so if you want to do that, that seems like a kind of a useful thing to do, but not the most exhilarating from a scientific perspective, but it can take its place there. So these biases built into AI tools, for me, is a big issue. And when you take it out into the real world, that there's always the Amazon example where they trained an AI tool to sort resumes, and what the tool did was basically give them resumes that looked identical to their current staff. Okay, so if you want to kind of move away from the past all the way down to the demographics of your organization, you have to be a little bit more thoughtful about how you would either build or use an AI tool. The last concern that I have about this is I worry a little bit about how you could ever get an AI tool to be innovative. So first of all, it doesn't think. It only knows what you told it, and it only values what you told were valuable. And so if you don't know what the next step is, then how in the hell can you train an AI tool to know what the next step is? And so stepping outside of what you trained it on, that to me is kind of the forefront of where AI is going to have to get to be really helpful in drug discovery. Because I can get a bunch of chemists, I don't need an AI tool, and I don't need to spend 3 years training it to say, what's the next molecule that you would make? But when I ask the question, outside of that chemical series, what's the next series you would make? That's a very, very hard question for a chemist to ask, because they can only rely on the molecules that they've seen in many respects, like an AI tool, right? So for me, that's the place to watch, is how are these tools going to actually have innovative step functions occur in drug discovery? And how are you going to get away, from this kind of shackles that you put on the tool because of the way that you train it?
And that's really interesting, and I hadn't thought of it that way. But it's rather different if you think about pathology or radiology, right? Because you're just showing the machine thousands and thousands of pathology slides. You already have the diagnosis from the patient. And, we can't, with the human brain, can't possibly see as many interesting patterns in those histology slides as the machine can, right? And so, for all we know, maybe the machine finds these little random squiggles or patterns that turn out to be predictive of whether a patient will respond to a particular drug. Or the same thing with imaging. Right? What are some special features that you see on an MRI or a CT scan that indicate to you how that patient's going to respond to therapy? Which means you have got to connect it, and I think brilliant of Roche to have bought Flatiron, because you got to connect it to the medical record. It means nothing if you can't connect it to the patient. You have to know how the patient responds to a therapy. If you do hundreds of thousands of cases, you should eventually be able to predict that. And that's where I think AI is the low-hanging fruit of AI right now.
So it's more in the hospital patient treatment setting. The AI tools actually can help the doctors.
I think so.
Yeah,
right now.
and I think, I mean, again, you know, we do millions of pathology slides on animals, you know, all the time, and you know, that is exactly the first place that we go. You go to the images because that's already, you know, been proven that you can do that. You can build a tool that can recognize images, you know, in any form if you give it enough images,
Sure.
right? And so for me, that, you know, and then, you know, all the way down to picking clinical trial sites, right? You know, you can, you know, AI tools, you know, in terms of which ones recruit or what are the characteristics of ones you can give them the data, and the tool can develop an algorithm that you don't know how the algorithm works, but it spits out a good answer for you. And so in many respects, I agree with you that is the place where you will see it first. Okay, it's going to come later in drug discovery because some of the problems that are inherent in the tool or the approach make it difficult.
Right.
So, but let me say, machine learning methods can, though, tee up for insight. And I think maybe a decent example of this was the work done within Google Accelerated Science looking at diabetic eye disease. And it ingested the tool, the algorithm ingested a whole bunch of data that were images of the back of the eye, and was able to easily call out diabetic macular edema. The interesting aspect of this is it seemed to call out very clearly 4 different patterns of diabetic macular edema. And that's very strange because to the human eye, this all looks like edema within your macula. And the 4 types were independent of the severity of the edema. But there was something about the totality of the pattern that were falling into 4 distinct groups. So it sets up for insight because it motivates you where you wouldn't have been motivated before to go look at those groups of patients and ask, is there something going on, like with the patterns within the cytokines, within some aspect of that patient? And there set the whole machine in motion to go down the road where you might get a very effective medicine.
And that was a beautiful example. Really, that was an amazing piece of work. You mentioned preclinical animal models, and that just brings me to another point, which is that, you know, we've cured a heck of a lot of mice with cancer with drugs that don't work in humans. So how do we bridge the gap between the preclinical animal models and a phase 1? And to me, it's going to require the generation, the production of fully human ex vivo systems that are faithful to what you see in humans. And that includes systems, and in cancer it's a little bit easier perhaps. You can make organoids from tumors, but that's not good enough because the tumor is surrounded by all of this hostile tumor microenvironment. So you got to be able to populate those organoids with vessels, with immune cells. You really want to see a tumor in a bottle, as it were. Recreate the whole tumor in a bottle by populating it with epithelial cells and fibroblasts and immune cells and tumor cells and endothelial cells, vasculature. And if you can do that for every patient, you can easily get thousands of little dishes of these organoids, and you can try combinations of drugs on them. And that will help us say what should go into a clinical trial and what shouldn't go into a clinical trial. That, to me, is a key gap in the whole system of drug development, is this abyss between preclinical animal models and what actually works in patients.
I think an area that this conversation makes me think of is actually how innovation works and what we call innovation. Is it more depth in an area where you have identified an area, something of interest, or is it, and I'm not contrasting them, I'm not judging which one is better, both are very important, or is it more breadth of discovering something new that's effectively highly unconnected to what you knew already? So if we listen to the conversation here, 2 things. There are so many other diseases than cancer that we need to be tackling and that are not in the line spot. And I want to bridge to bring into this conversation the reward system that this industry is based on, ultimately, and how we are incented to invest our time, our effort, etc., on the 31st PD-1 versus on trying to address neurodegeneration or treatment-resistant depression or other issues that are extremely complex to address. That makes me, I used to tell the organization this. I had small children that went to play soccer, and I watched them evolve. And in the first 2 years, you had the ball, and you had 22 players, or if they were in their second year, you had only 20 because the goalies stayed in their goal. Everybody around the ball, because that's what the action is. But as they evolved, you had 22 players covering the whole field because everybody had a different role and a different approach to what it is that they can add value to. And I think as an industry, we need to reflect on this, that we need to allocate enough resources to go more in depth in the area that we know are going to be productive, but there is a limit to that. When it was very interesting that the 5th statin became a very big drug, I predict that there is almost no chance for the 5th immuno-oncology medicine to make it. You could even start to talk about ethical challenges to take more medicines of the same mode of action that may have a marginal 10% benefit over the first one or the second one. So there is I think a decent disincentive at the end of the process to have duplication, while there is a huge incentive at the start of the process to have a huge duplication because you appear to be successful. I think we really need to identify ways now as an industry and as an ecosystem to drive more money, more investment, more brain power into the areas that we have not yet partially understood to the extent that, for instance, the oncology field has progressed. And the reason I say this is that immuno-oncology has been around for 30 years. 40 years, but like trying, trying very hard and failing and not believing, etc. And then it came up. And I think what we need is to make sure that many other areas have that level of committed investment of people, talent, and money to go there. And we need to find a way to create a reward, an unfair advantage to investment in those areas. That's how we will help patients better and allow the industry to become more productive and more impactful.
And Alzheimer's is a perfect example of that. The NIH spent more money on HIV/AIDS, which is also very important, than it did on Alzheimer's disease until recently. And yet 1 out of every 2.5 of us is going to develop Alzheimer's disease over the age of 85, which will take the budget. We have a $3.6 trillion healthcare budget in the US. $250 billion of that is going just for Alzheimer's disease patients. By 2040, it'll be over $1 trillion, and the healthcare system will collapse. Mm-hmm. So we have got to do basic research. The companies that have had the Alzheimer's trials, they've all failed. They've all failed. And we need more basic science to figure out why they failed. Is it amyloid? Is it tau? What is it that is driving Alzheimer's or Parkinson's or ALS or whatever? And this is, sort of a plea to this audience. You've got to support basic science because there is nothing to translate if you don't have basic science.
Completely agree.
The issue is because of the failure in the Alzheimer's disease space, so even the large pharma are not doing much research anymore, right? So what will be another thing is puzzling is that many companies, including academics and companies, they all have a lot of proprietary data. But right now we don't have a mechanism to share those data. So industry together, for problem like Alzheimer's, you probably not one single company can tackle. it requires probably ecosystem to collaborate, right? Figure out what exactly the cause of the Alzheimer's disease. But so far, there's no mechanism. Share all this data. That every company has a proprietary.
But I thought that several companies were getting together.
Yes.
Maybe I'm wrong, to share their data on Alzheimer's. Am I wrong about that?
Yeah, there's
a
several good consortiums.
Several consortiums
Yeah.
around this, and
We've had-
certainly in cancer, we share our data with many other cancer centers. But
You
you're
know,
right.
Alzheimer may be a different problem, or at least there's another problem in that, which are very slow progressing diseases where at the time of clinical manifestation, disease process, 90-95% of the process has already taken place and your room for improvement has disappeared. And I would say, if you go and talk to people, 95% of people will say, that's the problem. The reality is you need a medicine that you take at the age of 45 that's going to impact your issue at the age of 78. But obviously, that's impossible to do in today's world. And investing to identify predictors, biomarkers that can predict your risk of evolving to that state 20, 10, 15 years later, and therefore allow for clinical trials to be conducted and long-term studies to be done, and find ways by which regulators and payers allow for that to happen in order to prevent the catastrophe that Laurie was predicting, which I agree will happen. That's the solution, I think. And you know, it took an enormous amount of work by the NIH and its advisory board to drive towards the creation of large cohort studies that may, perhaps, if they are large enough, allow for the discovery of such things as long-term predictor biomarkers. And there's a number of other diseases in which this is relevant. Those are places where investments today will enable great improvement in productivity of R&D 15, 20 years from now, and we have to do it.
So I just wanted to point out that we have what we call the Columbia study going on right now, which is, in Columbia, obviously, but, there's this mutant population down there in Medellin, and so we know roughly exactly when disease onset or manifestation will occur, late 40s or so. And so when we ran that study, to make it at least doable, we picked 5 years. We picked patients who were 5 years before that and then dosed them. And our first, unblinding of that is in something like, 2021 or something like that. And we started the study about 6 years ago. And so this is one of the problems. If you want to go out and get out in front of the disease, which is really ultimately the ultimate way to do it, you have to run a study, that just in this particular case is 11 years long, to even be able to tell whether that mechanism has any relevance at all 5 years prior to disease manifestation. So this is a very difficult problem in this disease.
No, I agree, and we need to be careful that those patients are similar to the slow progressors or later onset.
This is an inherited mutation, right?
Yeah, this is an inherited mutation. That's right. So you have to make the assumption that the disease that they have is relevant in some way to the kind of wild type disease that is out there. And in many respects, so we knew that was a risk going into it. And our feeling was, look at this is the perfect kind of scientific situation to go before the disease appears, right? And at least ask 5 years before that, can you do something? And at least you're making progress there, because right now you can't do that with a wild type Alzheimer's patient, because you don't know 5 years before that they have Alzheimer's, even though they do.
Well, let me give you an example just for the ecosystem players to reflect on. My first 15 years in the world of pharmaceutical was in vaccines. And hepatitis B vaccine was developed as a first recombinant vaccine and approved. And one of the big questions at that time, in 1986, 87, 88, was, oh great, it seems to be protective against transfusion acquired or in short-term studies. Is this going to have an impact on hepatocellular carcinoma 10, 15, 20 years down the road? And it was an open-ended question. And this question, in fact, is true for every single vaccine that we produce. The key thing is that the vaccine was approved, studies were conducted, they lasted 10 and 15 years, and they have demonstrated that there was a very, very large protection against it, over 95% protection against development of hepatocellular carcinoma, particularly in a Western African study and other in Asia. And there is nothing can accelerate that. But the key is that actually an incentive was put and a regulatory environment was created that allowed for the industry to develop an intervention that had a short-term benefit, which could be a biomarker equivalent, and then a long-term demonstration of
Can I
benefit.
agree with that? I just want to say I completely agree with that. The industry's tackled, you reminded us, Moncef, the industry's tackled exactly this problematic situation before, where you're trying to prevent an outcome that's a couple decades out. And the solution for other situations like MACE events caused by blood pressure or cholesterol, they were all because there was a permission, in a sense, to approve based on a hypothesis on a biomarker that then the industry and the community at large spent a couple decades validating. And then once that happened, that was the bootstrapping necessary to get to the end. So, and then that's happening in Alzheimer's on the optimistic side. And biomarker-based approvals right now, if you go in with a legitimate story to FDA, that'll be okay. And so it can happen again.
And cancer is an absolutely terrific field to think about cancer vaccines in, right? Because we've already created a couple of therapeutic vaccines for melanoma. Now for glioblastoma, the new one is in process at Dana-Farber. But we would all love to see the day when we vaccinated our children and our grandchildren with a cancer vaccine. And you could start doing that looking at hereditary cancers, right? Where you know somebody has Lee-Fraumeni syndrome and a mutation in P53, you know they're highly likely to develop a cancer, or has the APC mutation, whatever. So developing preventive cancer vaccines against that could be measured in a reasonable length of time.
Okay, our time is up. Thank you the panelists for a very thoughtful discussion. I appreciate your presence here. Thank you very much.